Okay, so just let me know, screen is visible.
Yes, ma'am.
Okay, so yesterday we covered this, how to work with packages and how to use different functions from packages. Like we covered some built-in functions, built-in packages, and some packages from CRAN, some packages from Bioconductor. Like this BioBase we checked, your string R, and genomic ranges. So there is one most important package that is for data manipulation. We'll see that. So here, yesterday I told about to download this from CRAN itself, right? So library, you need to download and activate this package by using this install command and library command. Let me activate this package. So as told, data manipulation, what all things we can do. So here you can see the functions I have written. In manipulation, we can do selection for column like here we'll get the functions like filter, select, then there is, you can see, mutate, right, arrange, also summarize. So these are very useful functions for filtering rows and also on basis of column. So when we create any variable, arrange any rows or summarize any data, these all things we can do. So this is nothing but like cleaning process or pre-processing or filtering in bioinformatics. This is required, right? Even we can do a chaining operation, this pipe, for wrapping different functions. So here code is more readable and we can definitely reduce the need of intermediate variable, right? So because when we wrap one function with another function, so output is nothing but it is input for second step. Okay. Again, we can do group wise operations. Suppose you are working with different groups like genes or sample or any condition. So with help of dplyr you can use group by function, and that function allows you to group your data based on that variable, and then you can perform operations within that group only. Like suppose you want to summarize for any gene. Okay, so these all things we are going to see today, how we can manipulate the data, how we can do manipulation, data manipulation. Also in advanced part you can join different data set. Suppose like bioinformatic data you're working and you need to combine, so you can use left join, right join, inner join, full join. So by this way you can merge the data frame based on certain column, and this is again very important when you want to integrate the data from different experimental conditions or databases. So let's see, and one more thing is you can also integrate different packages. Like suppose you want to after this manipulation you want to generate a graph, then with this outcome you can use ggplot2 package for generating a nice chart or plot. Okay, so integration with other packages is possible. Clear? So let's here I have created one this small data frame like names, month name, and their days. So this name is a one variable, or you can say one column, and days is another column from this table. Table name is my data. Let me execute this. Here you can see five observation, two variables. Five observation means five rows and two columns. If you want to check data here itself, then you can call this variable my data. This is the column, or you can visualize this in new window like this. So you can add many rows here. Just I have added five rows. So now let's see how we are going to use dplyr to filter data. Okay, you can create this data frame first. Okay, now as we want to generate one filtered data, so let's rename or create a new variable as a filtered data. For which database or which data frame you want to use, that you can fetch it here and then apply operator. Then one by one you can use functions as written here. Not necessary to use all, whatever require you can use. Like suppose I want to select only few columns, or there are only two columns, so let's select one of them. or, or if you want to select all, then you can mention all names. Like here I'm selecting both names and dates. Now use this as input and then filter on basis of row criteria. So suppose I'm applying filter for days column. So as there are few numerical values, you can see, let's filter greater than 30 only. Now I don't want any other function, I don't want to apply, so no need to use pipe operator. That's it. Let's execute this. Okay, here you can see new object is generated, that's filter data with two observation, two variables. So what exactly that? Let's visualize this. Filter. Okay, so only those rows are there who satisfy this condition, who I mean true for this condition. Okay, so these two rows like March and December who are having value greater than 30. So this is the example manually created data frame we used. Now let's take data frame iris, which we already worked with in file handling, right? So this data we are going to use. How to fetch in data, use the which table you want to fetch, that name, and that's it. So after that you can check whether it is installed or not, or just want to check dimension, so you can call dim function. Okay, so this is our data. So we already checked this data having 150 rows, if you remember. Okay, let's see. There are some values in last column. Other columns contains only numerical values, but last column contains character value, that is nothing but species name. So if you want to apply any logical operator like greater than, less than, like this for numerical data, you can apply for only four columns. Other than, if you want to check like compare or check, so that time you can use last column. So let's try. All of you fetch this data, check the dimension, so we can work these all functions one by one on this data set. So first we'll use filter itself, which is, now you are very much clear with this. Okay, filter. So let's filter on the basis of length, or if you want to visualize data here itself, just to check column names, because we need to use column names whenever we want to apply filter and all. So you can call head function and check, or you can simply call names function to check only, to display only column names. Okay, so here we can refer this. Now let's call filter function. So to store filter data, here I'm creating filter iris, or you can create any name. Okay, so we want to filter from this original data, type it with filter, and here we need to apply filter. So when we type here data, we need to provide, so as there are sepal length, sepal width, petal and width and length and species. So let's take first sepal length itself. If you want to check what actual filter you can apply, so let's visualize here the values like 5.1, 4.9, and around 4 to 5 average, or we can check from here as well. The minimum is I think 4.3, where maximum is 7.9. So accordingly we can set some middle value, like here I'm fetching, I think S it's capital. Let's select first sepal length, set it for, fetch it only for greater than 5 values. Those rows only satisfied this condition that we want to fetch. Execute. So let's see, as the dimension, original dimension of the database is 150. Now let's check what is the dimension for filtered data, filtered iris. Okay, so here filtered 118 and 5. So because we have not selected any column name, so all columns are there, but only rows are filtered on the basis of given criteria. So out of 150 we are getting 118 rows who satisfy this criteria. Okay, even you can check head or tail to verify. So all rows are greater than 5. Okay. Now either you can use for order from here itself, or you can apply function to arrange your data by using order. So by default it is showing ascending order, but if you want to specify descending order, then you can use DESC keyword. So let's create one variable to store arranged data or arranged iris, fetch original data. Okay, now time to arrange data in ascending order. So apply function, as we have fetched dplyr package, install dplyr package, so we can fetch these all functions. You can see your name of the package dplyr. Let's fetch basics applier or arrange. So which column you want to arrange, that you can mention. Now let's use petal length. So that's it. Okay, there is no dimension difference because we have not filtered anything. So the 150 by 5 only, but still we can check. Oh, better we can use head function or tail, anything. So you can check this column petal length. Here it is. As it is in ascending order, only minimum value is there, minimum and so on. So let's change it with the descending by using DESC keyword, like this, execute, and then check head. Now you can see the maximum value at the top, that is 6.9 for petal length column. Okay, so this is order. Now you already checked this data set. There are multiple species available, I think. We're not getting how many species and what all names are there. But yeah, we can do one thing. We can group it together on the basis of name of the species. And then if you can see here, for Virginica, there are multiple values available. Like sepal length is 7.9. Also, there is 7.1. Then for in between, there is a very versicolor, then somewhere it is setosa. So we are not getting exact average sepal length. What will be the sepal length for this species, or sepal width, or whatever, like petal length, petal width. So what we can do, we can group together on the basis of species name, and then for that species we can calculate the mean of sepal or mean of petal or any of this column. Okay, so for that we need group by function and summarize function. So how to use this? We already use select, filter, arrange. Let's do summarize group by for same data set. Okay, so whenever we are doing group by on the basis of species, then only you can take a summary or mean, or like for that particular column. So let's create one variable to store this summarized data. Okay, so this name I'm providing. This is the original data set. Use this, and then let's apply first group by. So as we want to apply for species column, S is capital, I think. Yeah, species. So we are fetching that species, and now on the basis of species, we are going to summarize the mean for let's take sepal width or length, any one of this. So what we want to do, mean is not belongs to dplyr package. Mean is a base function. But let's apply mean for sepal length. Now, whatever mean we are getting, that mean, or let's do one thing, we can save that mean as nothing but our average mean. Okay. Now, for this whole thing, we can apply function summarize. Summarize from dplyr. Okay. That's it. Let's check the parenthesis. Here it is generated summarize iris having three observations and two variables. Two variables means two columns, three rows. So let's see what is the result by calling summary iris. Okay, so here we can see species. There are only three species in that table, and average mean for sepal length we are getting. So we can see here Setosa, versicolor, and Virginica, I think, having maximum length for sepal. Even we can check for petal if we want to change this with petal to check average mean value for petal length or width. Anything we can check. Okay, so here you'll get. So in this way, you can generate a summary for this database, like species wise for each variable average value. Clear? So this way you can manipulate the data, and with help of this data, suppose you want to generate a cluster or a scatter plot. So by using this data, you can generate a scatter plot. So you'll get to know which species are closely related or which is outlier. Like these all things you'll come to know. Okay, so in this dplyr we learn select, filter, arrange, mutate. Okay, mutate is not done. So mutate is what? Generating new column with help of existing column, right? So let's do one thing, apply mutate function, or let's generate a mutate iris. Here I'm going to apply mutate. First we have to fetch data, or if you want to apply here itself somewhere in arrange, then also you can apply. Just need to use percent greater than percent and then use mutate function. Okay, because whenever we are applying function, we have to use pipe operator. So mutate, we can do either we can do addition of any two columns. Suppose let's take sepal length and petal length together, and let's check what is the total length of that for that species. So I'm creating here a new column. Okay, and that new column is nothing but sepal length, arithmetic operator, petal length. That's it. Okay, let's execute this. Done. So this is the value. This means this is the column name and these are the average value. Or if you want to generate this in existing table, so here suppose I'm generating with help of percent greater than percent, enter and use this function. Execute and check head. So now you can see there are previously there, or let me execute dimension for arrange data. Okay, now it is 150 rows but 6 columns because this new column is added, and that new column is nothing but mutated column having addition of sepal. What we have added, sepal length, that is 7.7 and 6.9. So you can verify this. So this way you can use mutate, then group by, summarize. So dplyr package is very useful for data manipulation. That's why it is called as a grammar of manipulation. Okay. Now let's explore more packages which are specifically used for biological data. Let's open new. So here, first we need some packages. So let's first install some packages from Bioconductor. Okay, I'll write the name of package, BioString. Okay, please install this package from Bioconductor. Yesterday we did this. How to install? Just go to browser, type this name from Bioconductor. Just need to copy these 3 lines and then activate the package by using library function. As I already installed, so I'm initiating that. So how to work with this package, or what all things we can do using BioString. Sorry. So BioString is bioconductor package. Basically, it is designed for sequence manipulation. Okay, sequence in the sense DNA, RNA, protein, like amino acid sequence, any kind of sequence. So first we can do with help of BioString, uh, the simple string we already generated, that is nothing but DNA sequence, suppose. So like this we have generated, right? This is a simple string. If you check class for this DNA sec, you'll get character. But with help of BioString, you can generate a sequence. This is nothing but sequence only, this line number 4. But we can apply function like DNA string if we want to generate DNA string. Then for RNA, you can use RNA string function. Also, amino acid string. Okay, so here you can handle nucleotide, amino acid separately. And what all thing we can do, if we want to check some pattern, or pattern in the sense that is nothing but motif, we can do motif search or matching. Okay, even we can match patterns. There are some functions like match pattern, v match pattern, pairwise alignment for this all purpose. Then if we are looking just for substring, like any uh, in biological sequence, suppose you want to extract or manipulate any substring, so there is a package, or there's a we can say a function called as a subsec or replace at. Okay, this these two functions are there. Even here we use a single sequence, but if you want to generate a set of sequence, so biostring also support for handling large data set. So rather than writing string, DNA string, here we can use string set. The same thing, same function name, only set we can attach to the, like this, for everything, like RNA string set, amino acid string set. So this we can do. Even we can do sequence analysis. Like analysis means what? Simply analysis GC content. This is a very widely used. Even finding motive, translation, transcription, these all things we can do. Okay, even data import export, as we need FASTA files, FASTQ files, or any different file format if you want to import. So that we can do with these all libraries. So let's first create one sequence. Let's see. or we can rename this as a sequence 1. Here I am first creating simple sequence. Okay, so these are two sequence. Now let's convert this as a string, DNA string object. So for that we are using function DNA string. Here you can see from biostring there is DNA multiple alignment, DNA string set, string state list. So these many functions are available. So let's generate one sequence. Now for this sequence we can take input as our existing sequence, that is sequence 1. Now let's call this sequence. So you can see here this is indicating as a list, or why it is like list. Let's execute. If we execute sequence 1, which is simple sequence, you can see like this. But the same sequence if we generate with the DNA string function, then you can see there are 15 letters in this object. It is a sequence and these are the color notation. Okay, even we can call class function to check class of this, that is a package biostring attribute. You can see. Okay, create for second sequence the same thing. Done. If you want to change sequence, you can just change some. Okay, so these are two different sequences. We have changed the sequence. Now if we want to perform alignment to check how similar they are or what exactly the base is similar, so there's a function that pairwise alignment we can call. So to store the result of alignment, we can create your own variable and then call function pairwise. Here you can see from biostring there are functions pairwise alignment and alignments. As we have only two, then we have to provide your name, which two sequences. Could not find function. We fetch the function, right? Let me check. Okay, let me execute first. You can try this. Done. Got the result? Okay. Here it is showing our score is -9. Are you aware about this terms like alignment? Then how to check alignment, match score, mismatch score, then gap penalty, these all things, or what all algorithms are used to align these all sequences. If there are two sequences, then local alignment, global alignment, is all things. Okay, so in pairwise alignment and multiple sequence alignments, these both actually in bioinformatics are used for comparing sequence, or just if you are looking for similarities between sequences, then you can do this alignment, so global and local. So here in output you can see global pairwise alignment. So here global alignment is used. Global means what? End to end. End to end means entire length. For these two sequences it is checking, like start to end, right? So the pattern and subject which against that sequence we are going to check the alignment. So here this is a optimal alignment, means basically it is used to maximize the similarity score. In that you can check match, you can check mismatch, is there any gap, so these all things observed. And this global alignment basically using Needleman and Wunsch algorithm. This is very classic example for global alignment. When we want to compare sequence with similar length and similar structure, then basically prefer this global alignment. Then what is the local alignment? Local alignment means, suppose you want to check a region of similarity between two sequence, only region of similarity, and that allows gaps, that allows mismatch, these all things. So in local, there is a Smith-Waterman algorithm is used. Okay, means you're just looking for subsequences, and that subsequences with higher similarity score, not necessary to check from end to end. Okay, so when it is required local alignment. Okay, let me ask first. In blast is already done in your first model, right? So in blast, which alignment method is used? Blast tool? Anybody? This is very basic question. Or if you're not aware about these all terminologies, I suggest please read what is the alignment, global alignment, local alignment, different alignments, how to calculate the score, what is match, mismatch, gaps, these all things. Okay, then here we are just checking two sequences, that's why we are using pairwise alignment. But when we are working with multiple sequences, like or we can say global multiple sequence alignment, even there is a local multiple sequence alignment, so different algorithms are used for that. Like for global, or let me write here, multiple sequence alignment, for that you have to download MSA package from Bioconductor. Package name is MSA, small MSA. So if you want to allow align sequences for entire length, then specifically prefer global alignment. So in global, in multiple sequence alignment, there are different algorithms like cluster, they are inbuilt, no need to mention. Like here it is showing global alignment and all. So global cluster W, then there is a omega. Omega is also performing multiple sequence alignment with global alignment method. And in local MSA means local multiple sequence alignment. So there is a muscle, then I think mapped. These two algorithms are there which prefer local multiple sequence alignment.
Hello. Sorry to interrupt. So the line number 23, 22. I'm getting some error message.
Line number
22.
22. Okay.
Yeah. Shall I share my screen or I print the error
Uh,
message?
22,
Put it in the chat
22
window.
is not, we have not executed that. Actually, it is incomplete code because,
Oh, is it?
yeah, here I forgot to mention this. Like this is the code.
Oh, okay. Okay.
Yeah, so this variable hold that result. Okay,
Oh, we
because
are not executing.
function,
Okay,
yeah,
okay.
function, whenever we use function, we have to provide parameters or arguments. And in previous line which you are talking about, that 22, that is just a pairwise alignment, there is nothing after that. So
Okay.
yeah, these two argument we are passing for this pairwise alignment function.
Okay. Okay. Got it. Yeah.
Yeah.
Got it.
Hmm. Then okay, so let's see some more packages which we can work with sequence. Also, one more package you can download, that is from CRAN repository, called as a I'll here initiate. You can install that. Whenever we are working with sequence, that is important. That is seqin_r. Okay. Or if you want to install, you can just simply type command install dot packages and name of the package, that is seqin_r, and then activate that package. Here you can see all dependency packages. So now we are ready with seqin_r. Okay, seqin_r is basically used when you want to read or manipulate sequence, like you want to fetch sequence. Like suppose you just want to fetch a FASTA sequence. Okay, so that you can use with seqin_r. So let's fetch. If you have any sequence, then you can store that sequence in your current working directory. So we can directly fetch that from current working directory. Let me check. Oh, let me copy this. Okay, so I have pasted one sequence. Now we can work with sequence. Like first, let's create one variable to fetch that sequence. Like here I'm creating a sequence info. Call function read dot. Now here as we are calling FASTA file, so we can call read dot FASTA. You can see here seqin_r package having this function, and how to use this function you can just check these all things. There are many parameters are used but not necessary. We just provide the name of the file by using file parameter. Here you can provide the name. As I have fetched sequence for chr20, this is a FASTA sequence. Let me check first. It it is in process. This red dot means it's in process. Let me fetch first file, then we will use this file. Okay, so if you want to check file or what exactly the class of this, so you can call class function, call this variable. Okay, so this is list. Why list? Because there are multiple types of data. Like the first is greater than sign, then there is a name, then there is a sequence, right? So these are different data types. That's why it is showing list data structure. Now if we want to calculate from this sequence how many A's are there, how many like individual nucleotide frequency if we are looking for, right? So that you can do. But before that, let me show one simple sequence. Let me create one variable called as a DNSSEC, or you can create any name. This is a sequence. Let me execute this sequence. Now from this sequence, if we are looking how many A, T, G, C, so for that there is a function called as a table. Okay, so with help of table, you can first, as it is in character, so it is simple string. So first what we need to convert the string into character. Means this is a string. we need to convert into individual character. Then only we can count, like this A, T, C, G, separate buckets for each base, and that we can do with help of S2C function. And then table function we are going to create to generate a table of nucleotide frequency. So for that, let's create for the small sequence, then we'll create for the chromosome 20. So here I'm creating one variable to store this frequency as a frequency table. Then call first table function. First, we need to convert this into S2C, that is sequence, to character. Then provide the sequence for which sequence you want to calculate this frequency. Okay, like this. So we are getting this frequency table having 1 to 4, because we in our sequence there are only 4 bases ATGC. We have not added n ambiguous base. So let's check frequency table. Okay, clear? Now for this sequence, no need to use S2C function. Which sequence? Which is fetched through read.fasta, because it is in the form of the standard sequence pattern. So we can directly call table function to calculate table. So here you can see table is a biogeneric from that. And let's call, what is the variable name? Sequence info, which hold at chromosome 20 FASTA sequence. So as this is, I think, a big sequence, so it takes time. You can see this, it is in process. Let's wait for 2 minutes. Yeah. So in this sequence, you can see this is accession ID, then ATGC along with N. These are the count of individual base. Why we are not use this S2C? Because this is not a simple string. This is a string from Sekinar, or we have fetched this to package. So no need to convert this. In this way, we can do frequency count. Okay, now let's install MSA library, that is multiple sequence alignment. This, okay. Now how to work with this multiple sequence library? So first thing, let's create, as we want to work with multiple sequences. So let me fetch one sequence string, one vector. So this is amino acid string, vector of amino acid. And for that, let's create one name, or let me use here as a protein sequence or protein sec. First, we have to convert this into set of amino acid by calling function amino acid string set. Okay, and then provide this list. Let me execute this first. Some bracket is, I think. Okay, I'll give this link string to you as well in chat box, so you can perform. So now we have this string, and if we want to align these all sequences by using multiple sequence alignment, similarly, if you want to generate ATGC sequence like this, then you can just apply DNA string set function. Okay, so let's align these all proteins. That's why I'm creating one variable, that is align protein. Call MSA function. Inside MSA, we have to provide which variable hold all the sequences. So this is the variable where our three sequences are exist. And also there is one here you can see method. So if you want to provide any particular method for alignment, as we discussed here, there are different methods. So let's align, let's, here I'm using cluster W or cluster omega, anything you can use. Just check the alignment with help of align protein variable. So this type of output you'll get. So how to read this output? Can you see output? So in multiple sequences, here whatever the algorithm we provide, that is, you can see the command which we used, it is given, and how many rows and how many columns. Columns means how many amino acids are there, that is provided. Okay, this is align means alignment. Conscientious sequences means the sequence which indicate or that particularly derived from the alignment, so that represent most common. So here if you can check M which is common in two, so we can assume like this is the one which is correct, so M. But for second Li and third there is a gap, so means we're not conclude like what exactly the correct one, so that's why it is question mark. So which is most uh, frequently or maximum time appears, that thing we can say as a consensus sequences this line. Okay, let's, you can create sequence, like suppose this is my sequence. uh, Let me copy 3 times. Okay, just provide name 1, 2, 3. Need to execute these all three. So this way we have three sequences. Okay, these are not DNA strings, they are simple sequences, strings or character we can say. And for this also we can apply multiple sequence alignment. Suppose here I'm creating one variable alignment called multiple sequence alignment. As there are three sequence means more than one, so let's combine this by C. Let's fetch one by one. This is first comma second comma third. Okay, and here one more thing you can mention, the type of sequence. Like type you can mention either DNA or nucleotide, or you'll get recommendation. If you just check question mark or browse Wignity for MSA, you'll get all the details. So let me check with nucleotide. If it not works, then we'll try with a DNA. Okay, so it is showing value protein, DNA or RNA. So let's change this with DNA. Okay. So use default substitution matrix. Okay. And here we are not provided any type, which algorithm we want to prefer and all these things. So let's call this alignment variable to check output. This is alignment. Okay. So here you can see cluster 2.1. This is our command. And as they're all are same sequence, so in constant sequence, we are getting the same sequence as above three sequence. So cluster 2.1 is what? This is the software or you can say algorithm, and along with version we are getting like 2.1. Okay, which perform this multiple sequence alignment. Then there is a line call. Call is nothing but what command we used or what function we use here. Then MSA DNA multiple alignments with 3 rows and 44 columns. So this is nothing but how many sequence means how many rows and how many uh, bases, that is 60 columns, 44 columns, these all things. Then alignment, so alignment and then conscientious sequences. So this is conscience means which derived from the above three, that are nothing but most common between these three sequences. So every sequence is same, so we are getting same result. Okay, so this way you can use different bioconductor packages and different functions from that packages.
Hello, excuse me. One
Yes.
basic clarification, like align protein sequence, like why we are using AA string set, but we are not using the same thing for DNA sequences.
I'm just showing like what all different types we can use. If we want to use here DNA string set, we can create for uh, s- sequences by using DNA string set as well. But these are a without biostring package.
Okay.
Yeah.
And
This is
one more thing,
the same
while I'm
sequence you try with a DNA string set. Just
okay,
try, you'll get a result. In,
okay, and one more thing, that
yeah.
package for MSC, what is the command? Install MSC or like
No, no, MSA is from Bioconductor. So install MSA will not work. You have to go to a browser, or if you have this command for Bioconductor installation, BioC manager, then you can just use in that.
uh, via BioC manager we need to do, correct?
Yes, yes, yes. Not
I
CRAN, not CRAN.
Yeah, BioC manager install MSC.
Yeah, BioC manager, not directly BioC manager. You have to write. You have to write that whole command from Bioconductor. Like let me show this.
I'm
I
getting
have
some
a
error message. Yeah.
okay. Just let me show. Like this, some commands are there, right?
Oh, you need to write accordingly. Okay. Bio
This is not
manager.
required. This required. So here you just need to type multiple. Most of the commands which handles sequences that are from Bioconductor. So you have to work like this. Install like this. First line is for installation. Second line is for loading.
Yeah,
Like
while trying to install, I'm getting error message. While it is trying to install the dependency RCPP. Error in read RDS destination. Error reading from connection. I'm getting error.
okay, so whenever such kind of errors you are getting, you just need to copy that error, and there are many forums are available. When you put
Okay.
this all type of error in Google, now you'll get how to resolve that, like what all dependencies are required, and to install that dependency. Suppose there is any particular package dependency, then that package you have to install here in environment. Then only
Okay.
that tool will work.
Okay.
So
Okay.
that's
Got it.
why we need to click on that install along with dependencies. So that particular tool will install whatever dependency package is required for that to execute.
Okay. Okay. Yeah. Thank
Yeah. Okay, sir. Okay. Then see you tomorrow. I'll share these all scripts.